In advanced cases of diabetic macular edema, there is an accumulation of senescent cells in the retinal vasculature. This can cause loss of barrier integrity, vascular leak, and fluid accumulation. Dysfunctional senescent endothelial cells can also lead to the production of pro-inflammatory signals like IL-6, IL-1 beta, and TNF-alpha, which can lead to disease progression. Unity Biotechnology is developing senolytic agents that target the anti-apoptotic protein BCL-XL, which is overexpressed in senescent cells. In preclinical models, inhibition of BCL-XL leads to elimination of senescent cells while sparing normal cells. Studies in a diabetic mouse model demonstrate that BCL-XL inhibition reduces inflammation and improves endothelial barrier function. In animal models, UBX1325 triggers growth of healthy blood vessels and improves retinal function. In Phase 2 clinical study in DME, UBX1325 treatment led to a sustained improvement in visual acuity and stabilized central subfield thickness. UBX1325 is currently being assessed in additional clinical studies and may benefit patients with low vision.